So what you'll see in your lab manual is a DNA sequence at the very top. So we have an example DNA sequence, ACA, TC, and so forth, okay? And remember how DNA has one strand and a second strand. DNA is two-stranded, and then all the A's match with T's, all the C's match with G's. So therefore, you need to have one strand that you read and one strand that's simply there to kind of match up to it. So we call the strand that you're going to read the sense strand, because you're going to make sense out of that strand. And then you're going to take the other strand and you're going to call that the anti-sense strand. And so with our first code, what we're going to do is we're going to decode this DNA and turn the DNA into RNA. And what that means is you're going to take the sense strand, the ACA, TCT strand, and you're going to find out which RNA pieces match up to it. So if you have an A, what would match up to an A with RNA? Well, if you look at your code, if you have an A, it would match up to a U in RNA, because normally it would match up to a T, but in RNA you use U. So the first spot in RNA would be a U. And then if you have a C, what would match up with a C? Hopefully you said G. It's because G matches up to C's, and then you would get the next space in your RNA. So you now have a UG. What would match up with the A? Another U. With a T, we get an A. With a C, we get a G, and so on. So go ahead and finish doing the strand, and I'll slowly reveal what each one is. So now you have taken that DNA and you've decoded it and turned it into RNA. And the new RNA strand reads UGU, AGA, AUA, UGC, AAG. So now we'll take that RNA, and that RNA is going to be turned into a protein. So I've just recopied the RNA up there, and we're going to rewrite our RNA and translate that RNA using the second code. So if we look at the second code, what we can see is that it's a little more complicated. We're going to take the code in 3-letter bits. So for instance, if you look at the code, you can see that if you have a sequence of UUU, then the amino acid you would get is phenylalanine, which is represented by an F. In our RNA, we have a strand that says UGU. So we have to find UGU on the table. And you can use the table in one way. You can just kind of scan around and look for it. Or if you notice that the first letter is kind of broken up on the left side of the table with U's first, then C's and A's and G's. So if U is the first letter, look at the top part of the table. If G is the first letter of the 3-letter group, then look at the G's on the bottom of the table. So we are looking at UGU, so the first one is U, and so it's going to be at the top part of the table. Then if we look at the top of the table, we have different columns, and each column represents what's the second letter. So the second letter will be a G, so now we are at the upper part of the table with the G column, and so now we've got UGU, and then we can look over there, and we can see that UGU represents cysteine, which is represented by a C. Alright, so let's translate this. So if we take the UGU and find the UGU on the table, that will give us a C in the protein code. The next 3-letter code is AGA. So where would AGA be found? Well, it's a little bit, a third, 2/3 of the way down on, 3/4 of the way down on the table. G is the second letter, so it's over in the G column, all the way on the far right side. AGA, if you look at it, is arginine, and it's an R. Now you have to find AUA. So now find AUA. And hopefully you found which amino acid that goes to. It's isoleucine, which is an I. Now look for UGC on the table. UGC is a C, a cysteine again. Now look for AAG. Hopefully you found that AAG matches up with lysine, which for bizarre reasons is represented with a K. So now you have a protein, Crick. Francis Crick, the guy who figured out the structure of DNA. Hip hooray. So now you've taken your RNA and you've made a protein. Now what I'd like you to do is basically try some of the other ones. So for instance, here's another one. It's not in your book. First, take this one and turn it into RNA. So that means that you're going to take the sense strand and you're going to find the matching RNA that goes to it. So for instance, T's match up with A's. Oh, there's the whole thing. Then you're going to take that RNA and you're going to take it in 3-letter chunks. So you're going to take AUA, find AUA on your table, and figure out what AUA would represent in a protein code. I'll give you a moment to do that. And now look for CGG, and then look for GAG. Just take it in 3-letter chunks. And hopefully you spelled out my name, Irene. All right, your book has a couple others for you to try. And you can do those at your leisure. I will reveal the translations in a little while. But I will give you some time to do that. You might want to pause the presentation for a little while. If you try to see what transcription and translation look like in action, there's some fantastic videos found at the website dnai.org, and I will be showing you those videos later on in the course. But the reading the code section of that website has a lot of this information that is very useful in terms of thinking about what transcription and translation actually are. So your to-do list is to read the couple pages in your book, and then to do your traits survey, which is due next week, Monday. Homework number 9 is unscramble these pictures, and that is due Friday. Please try to unscramble the pictures on before Friday. And then optionally, take your time and go see the dnai.org website for some other cool animations and information about DNA. Thanks a bunch. See you on Friday.